Okay, well, welcome everybody. Thank you for sticking out the week. This is Module 5.1. We're going to be introducing a little bit of network modeling and using the Antimony syntax for doing model specification. Network modeling is a big topic. You could easily do a week or 2 weeks just on network modeling. And so I can only give you a very high-level introduction to the concepts of network modeling. So this is going to be an intro, and I realize that people will probably want to know more, and I will provide some links and references to things that could allow you to learn more. As always, I need to remind you that this is live streamed and recorded for future viewing on YouTube, and I want to thank our NSF funding sources, our prior NIH funding sources, and IU and OpenVT and Glimprint for helping us out. So today we're going to be talking first a little bit about biology. I wish I had more time to talk about the biology of networks. There's a lot of good material online. There are many good textbooks on this. I especially recommend Uri Alon's textbook. It's quite good. There also is an online Caltech course on network modeling. Herbert teaches a summer course on network modeling at University of Washington. That's very good. A lot of that's available online. We're going to talk a little bit about the kinds of networks that exist. We're going to do a very, very little bit about the mathematics of representing networks. Again, normally I teach a full semester course on this, so trying to do it in 2 hours is a little tough. But I'll give you a flavor, and then we're going to dive into how to actually implement networks with tellurium, antimony, and then also how to integrate them in CompuCell. And then this afternoon, we're going to focus on multi-cell integration of networks. So we're going to talk a little bit about networks. We're going to talk a little bit about antimony syntax. We're going to do 1 or 2 simple models of using antimony. And then I'll tell you about how you read and write antimony models from CompuCell. And we'll review that a little bit this afternoon. So you'll see some of the same slides twice because this is material that it really takes some time to consolidate. We may or may not get to some of the topics here, in which case I will encourage you to come back to the slides. There are longer versions of this course. I have a whole semester course on YouTube, which you could look at from the 2023 version of the CompuCell course, modules 5.1, 5.2, 5.3, and 8.4, which are available on the YouTube channel. Cover this in a little bit more detail. So if you want to go back and review this material in more detail, I certainly recommend checking that out. And there also are more exercises and slide decks, more detailed slide decks for that. The main resources for antimony, I would go online. There's a Tellurium Read the Docs by Herbert Sauro that's excellent. It has all the basics. Here's a picture of what that looks like. Definitely things to look at. Herbert has a great textbook, Systems Biology Introduction to Pathway Modeling. My slides essentially follow the order and thinking of his textbook. There are plenty of other good textbooks that take different perspectives if you want. And there's that tellurium cheat sheet, which is everything you need to know about antimony and tellurium on 1 page that I recommend. So, networks. Biology is full of things that we tend to represent as networks. Networks are a particular kind of abstraction. Not going to have time to talk about what's surprising about why network abstractions work. It's always a little bit surprising that they do work. However, I do want to be clear that networks are a very harsh simplification of reality. It's actually a bit surprising that they work at all. But there are many kinds of networks that we run into in biology. Chemical reaction and metabolic networks, the actual chemical transformations that create energy in the cell, that create the components of the cell. Signaling networks that relay information from the outside of the cell to the inside and back and within the cell.
Regulatory networks control gene expression in cells. What are called physiologically based pharmacokinetic networks, well, usually abbreviated PBPK networks, which talk about the absorption, distribution, metabolism, and elimination of drugs and other chemicals in the body, as those transport networks. And these all have typical timescales. Metabolism tends to be very fast, milliseconds to seconds. Signaling tends to take seconds to minutes. Gene regulation is slower because of transcriptional and translational delays. So turning a gene on or off typically takes about 15-20 minutes, sometimes a little more, sometimes a little bit less. Transport in the body is more complicated. The blood flow is pretty fast, about a minute or less, but a lot of these transport phenomena are slower on the scale of hours. There also are different kinds of abstraction that can be represented as network population dynamics models, for example, of viral infection, are a network model, although we don't usually think of them that way. And something called adverse outcome or disease pathway models are also network models. We're not really going to have time to talk about those today, but if you look in mathematical computational biology literature, you'll find a lot of that. There are a whole variety of networks in biology. We have to start somewhere. Signaling networks are one of the classic kinds of biological network. You have receptors on the surface of the cell. Those receptors could bind to receptors on target cells, or they could bind to chemicals floating in the environment. In either case, they typically will send signals through the cell. Those signals are typically mediated by kinases, which attach phosphate groups to cells, and phosphatases that remove phosphate groups from cells. And those signals then are amplified, filtered, combined, and relayed to get the cell to do something. There are a whole bunch of questions. For example, when the cell receptor receives a signal, is it destroyed at the cell membrane? Can it receive a new signal? And so on. Is there active transport into the cell, or is the receptor something that is basically a barrier that sends signals across the cell membrane? You can read a little bit about this. A classic example of a signaling cascade would be the famous MAP kinase cascade, where you have MAP kinase that phosphorylates MAP kinase that phosphorylates MAP kinase. And then MAP kinase does something. These are typical kinds of signal transduction cascades. And there are questions that you might want to ask. If we increase, for example, the level of growth factor in the cell's environment, does the cell proliferate or die? That's a signaling network question. Does changing the number of cell receptors for signal increase or decrease the cell's ability to migrate? For studying cancer metastasis, that's a typical question. If we have a mutation in a component of this network that affects the binding affinity, that would be for the receptor, of the receptor, does it change the strength or response of the network? Quite often, especially in diseases like cancer, something will happen where one of these steps is turned on all the time, constitutively active. In other cases, for example, with apoptosis, P53, something happens where one of these steps is turned off all the time, and that causes problems. And so then the question fundamentally for signaling is always going to be, does a change or mutation in that network change cell behaviors at all? And if so, does it change it in a way that leads to a disease state? So that's the kind of question you ask about signaling. The two most classic examples of signaling are kinases that phosphorylate, attach phosphate groups to other molecules. Often the phosphate is the active form of the molecule. The one without the phosphate is the inactive form, but it's not always true. Sometimes the other way around. Phosphatases take that phosphate group, dephosphorylate the molecule. And you'll typically get pairs of kinases and phosphatases that regulate the level of phosphorylated and non-phosphorylated versions of molecules. Complex formation is a little bit like chemical reaction, but we're not talking about a covalent bond here. We're talking about typically van der Waals or other kinds of reversible binding between molecules. Another thing that happens pretty often would be ubiquitination of molecules that marks them for degradation. And you can have a breakup of a complex followed by ubiquitination and then degradation of a molecular species. Sometimes the phosphorylation of a molecule triggers its ubiquitination and degradation. We often don't model active degradation, but it can be quite important. So phosphorylation, dephosphorylation, degradation control are typical questions that you ask about in signaling. If we're going to talk about gene regulatory networks, we have to remind ourselves of the central dogma in biology. You start out with DNA. The DNA is transcribed into RNA. That RNA is edited to make a messenger RNA. And then that messenger RNA is translated into protein, and the protein then has to be folded and then post-translationally modified. And so there are a whole bunch of stages that go from gene to protein. We tend in our diagrams to conveniently associate gene to protein directly. In our models, we often do the same thing, but in fact, there are a lot of intermediate steps. There is some minimal delay time once you start transcribing a gene till you get any protein at all. And those delays we typically don't model in detail. Sometimes it's important, sometimes it isn't. But it's important to be aware of these simplifications. Gene regulatory networks are often drawn using something that looks like circuit diagrams for integrated circuits. In fact, this abstraction is pretty harsh and doesn't work that well. Genes typically control long-term cell behaviors, turning things on and off. It's important to mention that the transcription factor regulation of genes is not stable. And so if you really want long-term turning off of genes in particular, there are other mechanisms that need to be used. Those could be methylation of the DNA itself, which prevents transcription, methylation of histones, which also reduces transcription. Acetylation of histones increases transcription. We know almost nothing about how acetylation and methylation happen. And so there's a huge amount of scientific uncertainty actually about how genes turn on and off. We typically model turning on and off of genes by transcription factors. That's part of what happens, but the main things are happening in ways that we don't understand scientifically yet. Many genes are either on or off in terms of transcription. That's not 100% true, but generally true. And so Boolean or Boolean stochastic models are often used to represent gene transcription networks. CompuCell has the ability to represent Boolean networks. I'm not going to be telling you how to do that today. I think you may get a little bit of that next week with MabBoss, but if you're interested, CompuCell allows you to do that either with MabBoss or with BioNetGen. When you're going to talk about gene regulatory networks, you might want to know what the rate of production of key molecules is in different cell types or between species. You often will do knockout or knock-in experiments where you say this gene is absent or not turned on, or this gene is always on instead of being on under particular conditions. You might want to understand how you get stable patterns of gene expression. Those are called typically differentiation. Those change during embryogenesis, wound healing, and cancer. Again, I will point out that while we typically assume that differentiation is due to transcription factors and it's induced by transcription factors, in fact, you cannot have stable differentiation due to transcription factors. And so a lot of the textbooks are simply incorrect
in the way they describe differentiation. We definitely often want to understand, especially if we're thinking about drugs to deal with cancer or drugs that might deal with the viral infection, for example, whether a drug turns genes on and off and how it changes molecular level. There are some genes which tend to be either on or off, and then the level of the protein is either on or off at a particular scale. There are other genes that are differentially expressed, where you get graded expression. That may or may not be due to particularly the transcription factor. Typically, when you get graded expression, it's not when a gene is transcribed, it tends to be transcribed at a relatively constant rate. However, the duty cycle, how much of the time the gene is on or off for transcription, changes, and that's how you get graded expression. And of course, you'd like to understand how the effects of a particular molecule being present or not change the behavior of the cell. That could be for toxicity, drug discovery, disease analysis, and so on. So these are all things that you might think about with gene regulation. Again, today we're going to be only talking about the mechanics of how to implement these things in CompuCell. But I want to give you at least a few minutes of background before we jump into the coding and ask some of the questions you might want to ask. Gene regulation typically has its own notation. The gene is typically drawn as a horizontal line with a little up and bent arrow to the right to indicate transcription. Here I have I is an inducer. The little lollipop arrow indicates that the presence of I increases the rate of transcription of the gene. In reality, that gene produces messenger RNA. That messenger RNA has to be modified, transported out of the nucleus, and then you have a ribosome that will translate that messenger RNA into protein, and then that protein has to fold and be post-translationally modified. We typically ignore that messenger RNA step, and we'll just say the gene that the inducer promotes the production of the protein. That's leaving out a bunch of steps, all of which could be regulated. It's amazing that these models work, but they'd work pretty well. There are a number of regulatory motifs that we'll run into. Actually, I should have edited this picture because these arrows have pointed ends, which is not correct. You have activation. Unfortunately, the arrow notation is inconsistent. Different people use different arrow notation. So you have to be ready to face that when you read the literature. A T-shaped arrow like that almost always indicates inhibition, but activation can be a pointed arrow, which I think is actually technically wrong, or a lollipop arrow. You can have gene repression, more of this thing coming in, the less of the gene you get. Multiple control, in which case you have to think about how those control signals interact. I'm not going to have time to talk about that today. It's a big unsolved scientific problem, although there's some basic rules for how to analyze it computationally. Gene cascades. I turn on the first gene, that gene turns off the second gene. That's typical. Autoregulation. My gene produces something that turns itself off. Self-inhibition is a very, very common motif in biology and has particular mathematical behaviors. Secondary regulation. In this case, this red dot is inhibiting the activity of a factor that turns the gene on. So the more of the red dot I have, the less of the gene I have. But the red dot doesn't bind directly to the gene, it binds to the thing that promotes the gene. And phosphorylation is often a very key component of this. The molecule that is going to either turn the gene on or off may be active or inactive, more active or less active, depending on whether it's phosphorylated or not. So one thing you'll notice about genes, and you'll see this in some of the slides, is that most genes have a background transcription rate. They're leaky. They're always turned on a little bit. We often will neglect that, make it zero, but there's always a little bit of leakage. That may or may not be important.
And again, if you want to do Boolean modeling, you can do that in CompuCell, but we're not going to talk about today. The transport networks, again, in some ways, they're a little bit simpler because everyone has more or less the same organs and the same blood flow. When you get down to microscopic details, you get a lot of heterogeneity, but at the main level of the body, unless you have something like situs inversus, it's going to be the same. And so the big physiologically based pharmacokinetic models often say, if I take a pill, how much of the chemical in the pill gets in the body? How fast does that happen? Elimination is done by metabolism in cells throughout the body, but especially in the liver. Elimination is essentially done by the kidneys and the liver through the urine and the feces, and the bile, although there can also be elimination through the breath and through sweat, for example. And so the high-level PBPK models are always about the same. You have compartments for the lung, kidney, heart, muscle, fat, and so on. The liver always has a special one. The gut always has a special one. And then you have blood, arterial and venous blood, that transport chemicals. And in this case, you want to know what is the time series concentration of a particular chemical in the body, especially in particular organs and the blood. After I've had a dose of a chemical, for example, take a drug, how long does it last? Where does it go? How do those things depend on my age, sex, weight, whether I have cardiovascular disease or not? What's the difference if I swallow the drug or inject it? Swallowed drugs tend to be very variable in the way they're taken up. How rapidly is the drug or toxin eliminated from the body? If it's a toxin, probably it's better to eliminate it quickly. If it's a drug, you might want to stick around for a long time. People modify drugs all the time to try to improve these profiles. Drugs are often very toxic in general, and so you might want to target them to particular organs, for example, cancer therapies. And so targeting is a big deal these days. And there are all sorts of interesting things you can do in terms of packaging drugs in liposomes or on nanoparticles, or combining them with molecules that they're cleaved by that release active forms in particular situations. So that's, I'm afraid, all I have time for to talk about the biology of networks. As I say, it's a huge topic. I encourage you to read more about it. There's a question in the chat. Can you run very detailed models on a personal computer? It involves a lot of cells and interactions. There are huge metabolic models with 20, 30,000 variables that are actually pretty well characterized. Those tend to use non-dynamic approaches called flux balance approaches. Those will run on a PC. One replicate of them will run on a PC with no problem. PBPK models of the pharmacokinetics will run easily. There are commercial packages like Certara that will run easily on your PC. So individual network models typically are not very demanding to run. They're fast. Now, if you want to run 100,000 replicas of a single network model inside every cell in your PC, in every cell in your simulation, then it takes more time. But almost no network model is so big that you can't run it locally as a single copy. So now I want to come back to network models. Classic example of a network model here would be metabolic network or simply chemical reaction network. And in fact, all chemical reaction diagrams are fundamentally network models, very, very simple ones, if I say. But the basic concept is a chemical reaction. This is a diagram of, let's see, ADH. This is a metabolic pathway. This is a glucose processing cycle here. Again, typically in our modeling, we neglect the details of metabolism that are going on in your mitochondria, just the way you neglect how the engine works in your car, unless it's not malfunctioning. But certainly, if you're interested in metabolic diseases, you have to model mitochondria in some detail. As I say, the way of diagramming networks varies a lot. There's a lot of inconsistency. One standard is called system biology graphical notation. There are other ones that are not compatible with it. In general, these networks all work together in the body. They don't work separately. You certainly have chemical reactions. If you didn't have chemical reactions, nothing would happen. You have activation and inhibition like signaling networks. These signaling networks often change gene regulation. So you have gene regulatory networks. And if you're thinking about the environment, you have transport, either active or passive. And so you have some transport model like a PBPK model. And so this was an example which we showed before from Wikipedia of cell cycle and cell death regulation. You have a variety of receptors on the surface of the cell. You have a variety of cell signaling networks inside of the cell. Those networks in turn turn on genes. So those are gene regulatory networks that will then control whether the cell proliferates or dies. So let's come back to the very simplest network, which is just a chemical reaction. Anything with an arrow in it is a network. Here, two molecules of ADP, adenosine diphosphate, are reacting to form a molecule, adenosine triphosphate, and adenosine monophosphate. So this would be a phosphorylation reaction. In this case, this is a reversible reaction, so it's got a double arrow. And this is a mass-conserving relationship. Essentially, the phosphate group is being transferred from one of these molecules to the other. I might have a slightly more complicated chemical reaction where I have two molecules of A become B. B can make three molecules of C. C can combine with A to make D. So these are typical chemical reaction formulae. If I draw the arrow in one direction only, it means it's an irreversible reaction. If I have a double arrow, then it's reversible. In either case, every reaction arrow should be decorated with a rate, the speed of that reaction. And that speed in general will only depend on the things at the root side of the arrow. If I have 2A going to B, the rate at which I produce B depends only on the amount of A. It might also depend on external factors like the pH, the temperature, the pressure, and so on. But the primary determinant is going to be the concentration of A. Similarly, going backwards, if I have C going back to B, the primary determinant of that rate will be C, the concentration of C. The net rate will depend on the concentrations of both B and C, but the forward rate is determined by B, the backward rate is determined by C. The number in front of a reaction, two molecules of A going to one molecule of ATP, two molecules of A going to one molecule of B, one molecule of B going to three molecules of C, are called the stoichiometry. That's a fancy name for that number of how many molecules are involved in the reaction. Okay. We now have the idea of a rate equation. Suppose we have A plus 2B going into 3C. Technically, and sometimes Word will turn things into italics when I don't want it to, the names of the species should be in Roman letters and capitals. The concentrations or amounts of those should be the same letters in italics. So A plus 2B goes to 3C is a statement about the molecules. A plus 2B goes to 3C in italics is a statement about the mathematics of the concentrations. So this is a rate law. VF is the rate at which A plus 2B goes to 3C. That will depend on the concentrations of A and B. We often will interpret rate laws as if they were continuous processes. In reality, every molecular interaction is discrete molecules. It's an agent-based system. And I can do those reactions, for example, using molecular dynamics or Gillespie formalism. We'll talk a little bit about Gillespie probably a little bit later.
depending on how fast we get through the material. If we assume that these are continuous processes and we actually represent A, B, and C concentrations as continuous variables, then we will often write these as ordinary differential equations, which are called reaction kinetics. And so here, dA by dt is minus Vf A comma B, and so on. Note that ODEs are not fundamental, and if I write chemical reactions in the form of ODEs, I can't go backwards. If I give you a diagram of this kind, you can always uniquely write the rate laws. The rate equations for ODEs. If I give you a set of ODEs, you cannot infer what the reactions were in general. And so it is actually never right to write ordinary differential equations to describe chemical reactions first. You always must write the diagrams, the arrow diagrams first. It is never correct to write MATLAB and to write ODEs directly. That's always wrong. So if we're going to go further, we need to have some hypothesis about how the rate of reaction depends on A and B. There are many assumptions about how you can have that happen. The most common one, which is true for simple chemical reactions at low concentrations, is called mass action. And it comes about from the simple observation that if I have 2A plus B going to 3C, and I have no A, I don't have a reaction. And if I have no B, I also have no reaction. And therefore, the rate of reaction has to go to 0 when the concentration of A or the concentration of B is 0. The simplest functional form that has that property is A times B, and B is raised to the second power. If I have 2 here in my stoichiometry, I get an exponent of 2 here. If you haven't run into mass action, I encourage you to read in one of the basic textbooks about how mass action works. Once I have a mass action rate law, for example, I can write A plus 2B goes to 3C at a rate, Kf, reaction rate, times the concentration of A times the concentration of B squared. This rate law is general. I can solve it with a Boolean network model. I can solve it with a Gillespie solver, stochastic solver. I can solve it with ODEs. If I turn it into ODEs like this, I can't go backwards. And again, if there's one thing you take away from this, you should never write ordinary differential equations to start with when you're going to describe chemical reactions or any kind of network. And putting them in MATLAB makes it worse. Okay. When you're talking about signaling and regulatory networks, you have a concept of activators and inhibitors. And this is why the pointy arrow shouldn't be used, because you don't want to confuse mass transformation with activation. Activators and inhibitors are not used up. They change the rate of a reaction, but they're not used up themselves. They're what are called catalysts. And so here I have a concept S1 is turning into S2. That's a transformation, so it has a pointed arrow. A is increasing the rate of that reaction. Here, the rate of production of S2 as a function of A goes up. As a chemical reaction, is often written shorthand. A promotes the production of S2. That is interpreted as nothing goes to S2 regulated by A, and that is a chemical reaction. Nothing goes to S2 with a rate that depends on A. Notice A is not used up. Yes, Carla and Clara, that's correct. Not saying that you should never use MATLAB, and I'm not saying you should never use ODEs. What I'm saying is that when you are thinking about a network, you should never start by writing ODEs. Because if I write a set of ordinary differential equations to describe a network, I cannot infer what the network was. If I draw the network and label it properly, I can derive the ODEs from that. But if I give you a network and ask you what the network, I give you a set of ODEs and ask you what the network was, that generated the ODEs. You can't infer that. That's actually an exercise I give students in my networks class. John Tyson, one of the great modelers, has published many, many, many papers of cell cycle over the years. The best ones, probably not some of the best ones in the world. Some of those papers, he only writes out the ordinary differential equations. And so I give the students those papers and ask you, what was the set of regulatory and chemical reaction and gene regulatory and signaling networks that generated the ODEs? You cannot determine it because there are many, many different networks that give the same mathematics. And so you can't go backwards. And so that's why I'm saying you shouldn't start from the ODEs. If you want to turn the network into an ODE later, that's fine. If you want to solve it with MATLAB later, that's fine. But if you publish the ODEs, you've lost all of the science and the knowledge that you used to create them. It's not interpretable. And so it's absolutely not good practice to start with ODEs or MATLAB because you can't infer you can't back out what you did. The knowledge that you used to create that is lost. The empty symbol is a question. What does this empty symbol mean? In many cases, especially in signaling networks, but also gene regulatory networks, there are many species of the cell that are present in large quantities that are used to manufacture S2. In this case, the empty symbol means we are not going to do the bookkeeping to keep track of the things that are used up to make S2. We're going to assume there's enough to make S2. Again, if we use the metaphor of a car, when I'm driving, I'm using up gasoline. However, I don't have to keep track of the use of the gasoline as long as I don't run out of gasoline and have the car stop. And so as long as there is gasoline in the tank, when I put my foot on the accelerator, the car will respond in the same way, independent of how much gasoline there is in the tank. So an empty symbol like this means effectively there's enough stuff to make S2 that I don't care about the stuff on the left-hand side. An inhibitory link is with a T arrow means that the more R I have, the less I produce S2. That's interpreted as nothing goes to S2 inhibited by R, which is written simply as nothing goes to S2 at a rate depending on R. So again, these inhibitory arrows don't show up on either side of the transformational arrow. They show up only in the rate. Again, there's a whole mathematics of this and there are whole textbooks about it. I can only allude to it today. Okay. Often, and this will be the last math I'm going to do explicitly here, often when we have this kind of regulatory system, we'll put our basically ignorance about how regulation works in the form of what is called a Hill equation or Michaelis-Menten equation. These are sigmoidal responses. If I have a to the n over k to the n plus a to the n, the bigger n is, the steeper my sigmoid is. I hid a slide which went into the details on this because I have time. K to the n is what's called the half maximal concentration. When a to the n is equal to k to the n, this thing will be a half. B1 is the basal rate of production. Vmax is the maximal rate of production. And this form is typically used for the regulation of the production of S2 by A. There are other forms one can use. There are exponential logarithmic forms as well. If the rate of production of if I explicitly write S1 goes to S2, and I'm not using mass action, I'm using a saturated form like Michaelis-Menten, then I would have Vmax S1 over K Michaelis plus S1. This says that as I get more S1, I have interference and I don't produce as much S2 as I'd expect, modulated by that Hill function. Again, I wish I had time to go over this in several lectures. I don't know how many people, maybe people in the room could give me just a show of hands about how many people have done network modeling before. Again, if you haven't done it, Herbert teaches a great summer school on it, both in person and online. We have recordings of a whole class that I teach on it that you could watch on YouTube if you like. That's all available for free. There's always a question of what values of Vmax and Km should be. I typically like to scale my values of concentration between 0 and 1. In that case, the Ks are 1/2. PBPK mathematics are transport equations. Those are based on transport rules. Those are typically linear. If I have blood going to some compartment in the body going to some compartment in the blood, the rate of transport is some transport rate forward times the concentration in the blood, and vice versa backwards. One thing that's a little tricky with PBPK is that whereas chemical reactions depend on concentrations, rates can depend on concentrations, and when you're actually doing transport, you have to work with amounts. And so then you need to know the volumes of the compartments so that you can convert back and forth between the concentrations of the chemical and the amounts of the chemical. If I have flow in the blood, I'm actually transporting amounts of the chemical. And so I have to know how much blood I have and the concentration to be able to do these. But you get these simple rules: change of concentration in the blood per time, this transport rate, the amount in the blood divided by the volume, minus the amount in the body divided by the volume. Again, these are just to give you a sense of what these kinds of models look like. The key thing is you always have rate laws. You have D by, if we're doing ODEs, if we're doing it stochastically, it's different. For this kind of transport, ODEs make sense. We'll have a rate, a first order derivative in time on the left, and a rate law on the right. Okay, now we're going to actually go into implementing things. It's taken me a little bit more time than I thought to go through the basics. But again, we're trying to, I hope that people have seen some of this before. I need everybody now to download Tellurium V2 from the student materials folder. That first link, the one that says Tellurium demo, will take you directly to this specific model. The second link will take you to all of the models. You're going to need more of all of those models eventually, but for now, we're just going to work with that first one. Somebody already ran it. You're way ahead of me. If it runs, you can put in the chat what you got. If you're having trouble locating it or downloading it, remember you have to put that zipped file somewhere where you can access it. Your desktop, maybe your folder. Yeah, people are getting it to run. Seems to be working for everybody, which is great. This is a model which is trivial. X is reacting to Y. Y is turning back into X. It follows what's called mass action kinetics. The rate X turns to Y is B times X. The rate the Y turns into X is C times Y. It's a trivial chemical reaction. Okay, this is the download information. Again, Tellurium V2 is a little program that allows you to run antimony models inside of CompuCell. You don't need CompuCell to run antimony models. You can run them in Jupyter notebooks and other places without using CompuCell, but it's convenient to learn them inside of CompuCell for our purposes. There are a bunch of exercises, sequential reactions, chemical equilibrium, Gillespie, and so on. We're going to explore one or two of these. We're not going to get to do all of them today. And I apologize. Last time we did this, some of the exercises were under 5.2, and I didn't renumber them. So you're going to see in class, we may do exercise 5.3, and it'll be numbered 5.2 here. I'll have on the slide the number of the exercise, but they may not correspond exactly to the number that I have on the slide. You're going to need to go to that demos folder as well. And again, these exercises 5.1, 5.2 are all in there too. You don't need to pull them right now, but finding them wouldn't be a bad idea. If you want to download them, that's fine. The reason I had a special link for the tellurium one is it's not numbered 5.5 underscore something. It's just numbered tellurium, so it's a little harder to find. Okay. Looks like everybody got it working. Is there anybody who needs help at this point? So what I'd like people to do is now open that code in Twit. And there's a lot of code there, and I'm going to ask you, I know it's hard to ignore code, but I'm going to ask you to ignore most of the code. And we'll come back and explain everything in that code and what it does. But for the moment, we're going to focus on the definition of the network model itself. And that is in lines 5 through 15 of the steppable. Maybe people can give me a thumbs up if you can get this open in Twit and find the steppable, PySteppable, and find the top of the code. It'll say type tellurium model here, model equals triple quotes, text, triple quotes, and then a line that says variables X colon red, Y colon blue. We are going to now do, in the next 20 minutes or so, a lightning introduction to antimony. And you'll see the names tellurium and antimony. And this is because Herbert Sauer at the University of Washington, who invented these tools, loves to give new names to everything. Antimony is the name of the model specification language. Tellurium is the name of the Python library that executes that language. And libRoadrunner is the C++ backend of tellurium that actually does the hard computing solving of the problems. And so if you see libRoadrunner, tellurium, and antimony, from your point of view, they are the same thing. However, antimony is the name of the language itself. Tellurium is the name of the functions that call and interpret antimony, and libRoadrunner are the solvers that actually implement tellurium. So people already have this open mostly. Remember that Player and Twit will both unzip the files for you. So if you've downloaded the zipped file, you can open them directly from Twit. You don't have to unzip it yourself. However, if you unzip it, that's fine. Just make sure you retain the directory structure. Line 18 controls what variables from antimony you're going to display and the colors you use. Here we're displaying two variables, X and Y. Notice if I look in lines 14 and 15, I have X and Y that correspond here. If I wanted to display three variables, I would add comma and then quote variable name colon color to line 18. And so now if I change my antimony model, I have to change line 18 to display it properly. The only lines we're going to be changing for the moment in our code are going to be lines 5 through 15 and line 18. The rest of the code we're not going to touch right at the moment. Well, I'll show you two lines we might change a little bit because the defaults may not be ideal. People have already gotten it running. The main thing you're going to get out of this, there are no cells in this model, by the way. There are no cells created at all in this model. Is going to be a scientific plot that's going to look like this. A lot of people pasted this into the chat already, which is great. The size of the dots we may adjust. The time scale we may adjust. The colors may be a little bit different on your screen. If there's a cell field, it's going to be blank because there are no cells. I'm going to come back and to this in a minute, but I said I wasn't going to play with the formatting very much, but I want to call your attention to two lines in your code. Line 25, it says self.add free-floating antimony model, bunch of stuff, and then it says step size. This step size converts the units of CompuCell, those Monte Carlo step units, to the units of tellurium. If you said that the CompuCell model was 1 minute per step, The tellurium model might be 1 second per step. In that case, you'd have to set a step size of 1/60. In line 36, self.plot window.add plot, style dots, color equals variables var, size equals 8. If the little dots on your screen are too small when you're displaying things, you could make that number for size a little bigger. That'll make the dots bigger. Do people want me to show you that in the code itself? I can go and do that if you want. Let me do that. So here is the model specification. Here's a list of the variables I'm going to plot. And if I scroll down to here, line 36 for me, I have the size of the dots I'm going to plot. If the dots on your screen are too small, you could make this say 8. That will change the size of the dots in the plot. You can try here. I have step size is 0.01. That's actually stretching out the antimony model quite a bit. Look in line 25, maybe type in the chat, is the step size 0.01 or 1 or something else for you? 0.01. Okay, that's pretty good. You can try seeing what happens if I make that 0.05, make it half as big, then run it again, and you'll see that you'll get half as much change per unit Monte Carlo step. So the smaller this is, the less time we do in each antimony step. And so again, if my internal antimony time were 1 second and my Monte Carlo step were 60 seconds, I would have to set step size to 1/60. So CompuCell doesn't do that conversion for you. You have to figure that out. So if you're using an existing antimony model where its step size is 1 second and CompuCell had 5 minutes as its step size, then you'd have to make this number 1/300. Has anybody tried changing either the step size or the size of the dots and see what happens? So if you have, why don't you put that in the chat for us? Yeah, somebody says make it smaller, it becomes slower. So I'm going to just tell you this. It may or may not work very well on your computer. But when you have a scientific plot, if you right-click on the scientific plot, there are a variety of options, some of which will crash player, but one of which is export, and that should still work. And if you hit export, you can either save the image of the screen there, or you can actually save the data in the plot as a CSV file. There's a question about what are the step sizes for antimony and CC3D. When you define the models, you have to make those decisions. Those are not intrinsic to either antimony or CC3D. When you do your modeling, you will make a decision that in my antimony model, 1 unit of time represents 1 second or 1 minute or 1 millisecond. And similarly, when you define a CompuCell model, you'll say 1 Monte Carlo step unit of time represents 1 minute, 5 minutes, 10 minutes, 1 second. And so those are biological decisions. CSV is actually a table. It's actually all the data in tabular form, X, Y. I don't want to play with that very much now, but exporting data with file opening and closing is something we're not going to have time to talk about this week. And so sometimes it's useful to actually be able to grab the data from the CompuCell without having to write files explicitly on your own. So that can be useful. I'm now going to go over antimony syntax. The nice thing about antimony compared to MATLAB or Mathematica or other systems is that it's extremely simple. The downside is also it's extremely simple. We're not sophisticated the way MATLAB or Mathematica are. We can only do certain limited things. But for our purposes, it's enough. Okay, so what do we need to know? First, antimony models are strings. They are Python strings. We use triple quotes to specify the string, and it could be triple single quotes or triple double quotes. They have to be the same because we're going to have carriage returns. And in Python, if we want a string defined with carriage returns, it has to be defined inside of triple quotes, not single quotes, but triple quotes. The second thing is that the network specification is text, simple ASCII text. The third thing is that you can have comments in antimony. Double slash or hash specify comment. Double slash equations. This is where my equations are. Double slash parameters. This is where my parameters are. When you're doing the simple exercises here, you may not want or have time to type comments. If you are going to be building publishable models, you should have documentation about the meaning and reason for everything you're doing, in which case you should have comments. Semicolon specifies breaks between sections, and it works also instead of a new line character. And if anybody knows C++, you'll recognize where the semicolon comes from. You will see at the beginning of some lines, like lines 6 and 7, something colon. Colons represent names. They assign names to rate laws or events. And they have an advantage that they actually give you a variable. This assignment creates a variable which will return the value of the rate law. So if I want to know what is b times x, J1 equals b times x. If you decide to name things, the names have to be unique. If I name two things J1, it's going to ignore the first one. That's an easy way to make mistakes when you combine code from multiple places. Antimony was designed to write chemical reactions. And so the fundamental thing that you write in antimony are arrows. X goes to Y, Y goes to X, and rates, B times X. Notice the semicolons. And so if I have the chemical reaction X goes to Y at a rate B times X, I type X dash arrow Y semicolon B times X. The great thing about antimony is that every arrow you have shows up as an arrow in your code. Antimony is what is called the declarative language. Declarative languages are different from procedural languages like Python. They're compiled once on load. And in general, with a few very specific exceptions, the order of the lines in antimony doesn't matter. If I reverse lines 6 and 7, it doesn't change anything. If I reverse lines 10 and 11, it doesn't change anything. That makes things easier to combine. It also sometimes can be a little bit confusing, but it's typical of declarative languages. If people have, I don't know how many people have ever used a declarative language as opposed to procedural language. Something like MATLAB or Python or Mathematica are classic procedural languages. Declarative language, SBML is a declarative language. Arrows create rate laws. In antimony, typically we solve these by ODEs, but they can be solved by other solvers as well. You can solve by Gillespie. So the great thing about writing this arrow diagram is that I could solve this as an ordinary differential equation, or I could solve it as a Gillespie stochastic solver without changing this specification of the model. That's why it's so powerful. I can't do that in MATLAB or Mathematica. Again, naming a reaction gives me access to the value of the rate. J1, X goes to Y, semicolon B times X. J1 will have the value B times X at any time. Okay. That's the basics. Notice that one chemical reaction, X goes to Y, affects two different ODEs, because this is the rate at which X is converted to Y. So I reduce the amount of X at a rate B times X, and I increase the amount of Y at a rate B times Y. Y goes to X at a rate C times Y, increases the amount of X at a rate C times Y, and decreases it at a rate C times Y, Y at C times Y. So arrow diagrams are also more compact than ODE expressions, as well as being more specific and more portable. Antimony handles stoichiometry, complex numbers of molecules, automatically. If I write E2, 3A plus 5B goes to A plus 2C plus 9D at a rate K times A cubed plus times B to the fifth, it will automatically subtract and add the correct numbers of A, B, and C and D in the ODEs. If I do this with ODEs, I have to keep track of all that stoichiometry by hand. Antimony does it automatically. One small thing to worry about is that exponentiation in antimony uses the up caret, and it, of course, in Python, it's the star star. And so you have to remember antimony is not Python. The separators, the semicolon, other things. Indentation doesn't matter in antimony. And so the one thing that people do trip on quite a bit is that the exponentiation operator is different. So you have to remember exponentiation is up caret. As a declarative language, antimony requires you to specify the values of all parameters. It will throw an error if you don't do that, and the initial values of the variables. As a matter of good practice, you should have a comment at the end of your parameters and your variables explaining what the units are. I haven't done it here because this is a toy demo, but in fact, we should say the units of B are moles per second, moles per second squared, moles squared per second. So it's very important to keep track of that, because when you do unit conversion, if you haven't written it down, it's confusing. Antimony will tolerate not specifying initial values of variables, but it's strongly discouraged. It will crash if you fail to specify initial values of parameters. Somebody asked about nothing goes to A before. Antimony will let you write dangling arrows. I can write arrow to A, which would be written E1 colon arrow goes to A semicolon K. This would be the creation of A at a constant rate. dA by dt equals K. I can also have something disappear if I don't care about what happens to the thing that disappears. E2 colon A goes to nothing at a rate K times A. If you like, arrow goes to A is equivalent to just writing dA by dt. It's equivalent to writing an ODE, if you want to think about it that way. It has the advantage, though, that I don't have to solve it as an ODE. I can solve it in other ways as well. There's a question about parentheses. In general, the standard operator precedence works. I tend to put in a lot of parentheses when I write these things because I don't want to accidentally get hit by having the parentheses not be what I expect. Unlike Python, antimony treats everything as a float. But I still recommend that you write 1.0 for things to remind yourself that it's a float. If I have multiple reactions that involve the same species, nothing goes to T, T plus V goes to I1, then in the net rate laws, antimony will automatically sum them up, which is also pretty convenient. In general, it's, while you can combine things together a lot, I think that it's better practice to separate creation of things, destruction of things, and transformation of things in multiple lines. So you have the freedom to create as many lines as you like for each of the rate laws that you want. Antimony has a bunch of other capabilities. I have to get us to actually using it a little bit before we run out of time. Antimony has a bunch of other capabilities. You can write ODEs directly with the accent mark. I don't recommend doing that, but you can if you have to. That's functionally equivalent to writing colon arrow to T. Assignments made in antimony are evaluated only once when the antimony model is loaded. That can be confusing because if you have something like K2 equals 3 times K1 and I change the value of K1 later, the value of K2 doesn't change. There is a special kind of equal sign in antimony colon equals that says whenever I update the value of K1, also update the value of K2. So antimony has two, just the way Python has an equal sign for assignment and equal sign for comparison. Antimony has an equal sign for evaluate at load and another equal sign that says keep updating. I have slides about that, which I'm not going to have time to go over today. Antimony also allows you to do events where something happens at a specific time or under a specific condition. Their syntax is quite simple. I can name the event, don't have to, at, and then a logical expression, colon, and then something that happens. At time greater than 15, A equals A plus 15. That at time 15 adds 15 to A. Notice that this doesn't happen every time after time 15. Events trigger once when the trigger goes from false to true. So this happens only once. If I wrote E1 colon at sine of time greater than 0.5, B equals B plus 0.05, then every time sine of time goes from being less than 0.5 to greater than 0.5, it'll happen again. And so you can have triggering on values. I could say, if B is greater than A, then do something. If A is bigger than 15, then do something. You can build very complex logical rules here. There are a couple of complications. If I have two triggers that trigger at the same time that affect the same variable in the same way, only one of them will trigger. And so that's a little bit of an annoyance. We're not going to run into that today. So now we're going to actually do some antimony cutting. You started out with this code. X goes to Y, Y goes to X. Parameters were here. First thing I want you to do is change these values of the parameters. We'll just take 2 minutes to do this. Change the value of B from say 0.01 to 0.05, or the value of C from 0.1 to 0.5, or the value of C from 0 to 0.01 to 0.1. Try that and see what happens. Just to make sure that you're able to change values in antimony and have things work. Okay, I see a lot of people exploring. If I had more time, I'd talk about chemical equilibria and how those equilibrium values depend on the values of B and C. And this is an exercise that could do that. So this is the result. The equilibrium value of X and Y is C over B. So you should see that that equilibration curve changes. The next thing that I want you to do is to actually type a little bit of an antimony model. This will give a couple of minutes for. We started out with X goes to Y, Y goes to X. Here's what I want you to type. I want you to type A goes to B, B goes to C, C goes to D. Rates KAA, KBB, KCC. You're going to have to change the variables. If you change your variables here from X to Y, X to A, you'll have to change the variable in line 18 from X to A. You'll have to add a plot for B and for C and D as well. Give them color names. All right. And remember that you'll have to change the rates.
Here I called it Ka, Kb, Kc. If you want to call it something else, you'll have to change the parameters here. If you don't give initial values for the parameters, it will crash, and you should give initial values. If you start out with all A, B, C, and D 0, you're not going to get anything happen. But you might start out with A equals 10, B equals 0, C equals 0, D equals 0. When you get it working, put it in the chat. Okay, there's some good questions in the chat. Do spaces matter in antimony? No, spaces don't matter in antimony, neither does indentation. However, capitalization does matter. Somebody says if I change the rates from B times A and C times B, don't use, even though capitalization matters, it can be tricky. Don't use the parameter names B and C. Use something like Ka, Kb, Kc. Don't use A, B, and C. It's dangerous to have the rate be lowercase a and the value be uppercase a. Don't do that. For people who are still working on it, I'll leave it up. If people have it running already, I recommend there's some quite interesting things that happen as you change the values of Ka, Kb, Kc. I recommend trying 1, 10, 100, and 1.1.01 as values of Ka, Kb, and Kc. If you really get stuck, exercise 5_1_2_ sequential reactions is the code with this change in it. Somebody said they had a CVODE error. If you had a CVODE error, you didn't type this correctly. A CVODE error almost certainly results from a divide by 0 somewhere in your code. So you should get something that looks like this. Depending on the time scale you use and the colors you use and the dot size you use, it may look a little bit different. Depending on the initial value of A you use, it may look a little bit different. I'll leave this up so people can copy it if you haven't gotten it working yet. Again, you don't need to name these, so you could save time by not typing the names. A goes to B, semicolon, Ka times A. B goes to C, semicolon, B times B, Kb times B. C goes to D, semicolon, C times D, C times C. Ka, Kb, Kc initially are all 1. A equals 0, 10, B 0, C 0, D 0. Variables A colon red, B colon blue, C colon green, D colon orange. It would be reasonable to ask me what colors are allowed. I can't tell you. You can try them out. The normal colors work. Red, blue, orange, green, pink, I think works. I don't know whether fuchsia works. Remember, capitalization matters. So capital K times A is not the same as lowercase k times A. That's a big source of errors. If you have very long code, like hundreds of lines of this, debugging it can take a while. So the solvers, I'm not going to talk today about how you set the solvers. There, I'll show it to you, but this is something you'll look up on Herbert's website. There are lots of controls for the numerics of the solvers. This particular model is a very simple one, and so it shouldn't require you to adjust the solver conditions. But when you run complicated models, sometimes you have to adjust the solver tolerances and time steps. Kaya says, is it possible to use VS Code instead of Twit? Of course, you're welcome to use any editor you like. VS Code has a lot of advantages. There are code snippets available for Visual Studio, although I haven't used them. We'd love to have somebody, if we were going to do this again today, we would probably use VS Code as our primary editor. The advantage of VS Code is it gives you things like LLMs built in. The disadvantage is that it's a little more complicated to install and use. And so for learning purposes, we typically use Twit, and Twit does have the built-in code snippets. But by all means, if you like to use VS Code, use VS Code.
I'm really pleased to see people helping each other in the chat. That's really helpful. So there's some annoyances with antimony, and I encourage people to practice it. As I say, when I teach antimony, normally I have a whole semester to do it, and today I have 2 hours. So that limits things a little bit. Antimony has a lot of reserved words, and if you use those reserved words as variable parameter names, it will crash with uninformative errors. Because antimony is a string, it doesn't typically tell you where the problem is. It will just crash. That's an annoyance. In particular, the words time and gamma are reserved words. So don't name parameters simple things like time or delta or gamma. I strongly recommend, that's why I use Ka, for example, that you always give parameter names something like gamma 1 or delta 1 to avoid overlapping on reserved words. If you have missing or extra parentheses, mismatched parentheses, or improper separators, you left out a semicolon, tellurium tends to die, and it tends to die in unhelpful ways. If you're going to debug, my suggestion is comment everything out in your antimony string, and then start uncommenting from the top, 1 line at a time. However, when you're doing that, remember you have to uncomment the variable and parameter initializations, because if you use a parameter without initializing it, it will crash. So comment everything out, uncomment the variables, uncomment the parameters. Make sure that those work. If you uncomment the variables and it crashes, you had a typo in a separator or something like that. If you uncomment the parameters, same thing. No rate loss. And then uncomment the rate laws 1 at a time until you find the one that has a problem. Usually it's a divide by 0, or you've got the name of a variable parameter wrong. Usually uppercase, lowercase confusion. Events also, if you're using them, should be uncommented 1 at a time. Okay, the next thing I'm going to talk about, and we're really going to do this later, so this is getting ready for the afternoon exercises, and I will go over this again in the afternoon, is how you use antimony in CompuCell. The integration is through Python, and we have 2 options. We can load antimony models into the entire simulation. They're called free-floating models. That's what we've done now. Or we can load antimony models into individual cells, which is what we're going to do if we're modeling gene regulation or signaling or metabolism. All antimony parameters and variables are automatically available as dictionaries. And so if you're not a Python person, brush up on dictionaries. Everything in CompuCell uses dictionaries. That's how it works. So the dictionaries are critical. Time stepping, you have to do time stepping of the antimony model. And you have to decide whether you're going to use the ODE solvers or the stochastic Gillespie solvers. Remember to use the code snippets. A little bit unhelpfully, the code snippets are hiding under the heading SBML solver rather than under the heading antimony. And the reason for that is that CompuCell allows you to write SBML models as well as antimony models. Probably we should have said network solvers here, but because when this was done originally, we were only supporting SBML. It says SBML. And so you're going to see things that say SBML when you really mean antimony. If I pull down that TWIDIT menu, SBML solver, you will see add antimony model to cell IDs, add antimony model to cell types, and so on. I can show you that in a minute. If I look at the code that I was just showing you, you'll see in line 30, add free-floating antimony model, model string equals model. This is the name of the string I gave in line 4. Model name, that's how I'm going to refer to that model from CompuCell. And then the step size, that time conversion. if I'm going to plot information, here I'm plotting a data point. And I'm referencing the value of a variable, self.spml.model. And that's a dictionary entry. Yes, you can load, so there's a question, can you use SBML in CompuCell? Absolutely. I could define a model string that was an SBML string. I could load the SBML string. I can read SBML from files. And in previous years, when I had more time, I would show you how to download SBML models from biomodels, pull them into CompuCell, and run them. So yes, you can use SBML. SBML is not as human readable as antimony. That's why for our purposes we're using antimony. By the way, there's a single command that will convert antimony to SBML and vice versa. So if you have antimony and you want SBML, you can actually, there's a website that will convert it for you. ChatGPT will convert it for you at the moment as well, and vice versa. So they're interchangeable. Okay. Again, we could load SBML models, we could load cell ML models, we could load MAMOS models. They're all possible. So if we're going to use antimony models in CompuCell, we need to define the antimony model. In our case, we made it a string inside of CompuCell, but we could read it from a file too. Add free-floating antimony model, loads a single copy of the model that runs globally. Sorry, the model is loaded from model string. The name of the model is model name. And CC3D will refer to those models based on whatever you said model name was. Self.spml.model. Notice that it says SBML here, not antimony. Once it's loaded, it calls it SBML whether it's antimony or not. That's a bit confusing. Once I've loaded it, typically in the start function, when I step my model in the step function, I do self.timestep SBML, and that will timestep all of the antimony models together. I can have different antimony models with different step sizes. So if I want one model to run faster in one cell than another, when I load it, I can change the step size. But when I time step here, it all steps everything in synchrony. There are a bunch of options for that solver. There's an option, self.get SBML global options, that'll return all of the integrator settings, tolerances, and so on. So if you're really getting C-vote errors, which are conversion errors in the solvers, you can change the settings. I'm not going to talk about that today. Nothing we do today should give you that. But if you're writing very fancy models, it can happen. Again, self.spml.model, whatever that model name is, square bracket variable, returns the value of the antimony variable parameter. I can read and write it freely. I can say some value A equals self.spml.model quote A, or I could say self.spml.model quote A equals some value. So reading and writing, provided I remember the syntax, self.spml.model, square bracket, name of variable or parameter, I can read or write it freely. I don't have to do anything fancy to go back and forth between antimony and CompuCell, or for that matter, from SBML or CellML. Now, J1, which is a named rate law, I can read that, but I can't write to it. I'm not allowed to write to it. Okay, so a summary. Self.spml model here is because I called the model. If I gave the model name hello, it would be self.spml.hello, square bracket variable name. And that's all you need to know to use antimony inside of CompuCell. So what I want you to do now to practice this is take the code you were just running
And in line 32, add self.sbml.model square bracket quote Ka close quote equals 2. If you don't remember how to do that, pull down the SBML code snippets, find set SBML state for free-floating model. And that will paste the code in that you need. You'll have to edit the name of the model and the variable name. Can people do that? So you're adding one line to your code, line 32. All of this code is in the Python steppable. It's not in the XML. The most common cause of a C-vote error is when you have a divide by zero and a rate law. If you want to change structure of models, it's a little tricky to do that in Antimony. Events are a good way to do that. In general, changing the expression of a rate from something like linear to quadratic, the best way to do that would be to say k times a to the n and make n a variable, which you change. What you don't want to start doing is having to overwrite the strings themselves. I've had people who write complicated string editors in the Python and change the antimony model string by hand using things like grep and awk. It works, but I'm not, or the various Python functions that are string editors. If you want to do combinatorics, you can do that, but I wouldn't encourage you to do that unless you have to. Somebody says it doesn't make any difference. Doesn't make an enormous difference. It'll change the values a little bit. You can change the values of variables as well. You could change the value B, C, and so on. And maybe during the break, people can explore that. You're going to have to change the model name and the reactant name to be the correct things for your model. You might very well, somebody asked, can you change the parameter values iteratively? Yes. You can do all of these things. Whether you want to do them is going to depend a little bit on what kinds of models you're trying to develop. Again, I'm showing you the basic mechanics of how to do these things. And this afternoon, I'll show you an example where we really use it. But this is something where these models can become extremely complex. The point here I think I want to make is that the syntax you need to build a very complex model is very simple. As I say, to learn Antimony, you need to learn 5 concepts only. Everything about the language fits on one piece of paper, which I like. If you're stuck, you can download this exercise 5_2_1 Antimony in Python. The last thing I wanted to do, and I guess we're out of time, almost out of time, we'll see if we can get it, is Gillespie models. As I mentioned, you can do stochastic solvers instead of deterministic solvers in Antimony. And that's pretty powerful. One thing you have to worry about is Gillespie models count number of molecules. Typically, the concentrations that we use are written in micromolar concentrations. Gillespie, you actually have to use numbers that are counts of molecules. And so you have to be aware that there's a conversion factor there. In this case, we arbitrarily set A equals to 10, so that will work. It's also a much slower solver. But if you want to do a Gillespie model, you would take in the solver, when you load the model, you get rid of the word step size. You don't have step sizes in Gillespie. And you replace it with comma integrator equals quote Gillespie. And the step size actually has to be corrected. So I want you to try this. This will be the last thing we're going to do. If you don't have time to do this now, I would suggest you try it during the break. I'm not going to have too much time because we're almost out of time. In line 33, integrator equals Gillespie instead of step size equals 0.1. And if you want to be fancier, you would write actually, well, we're not going to do this one. This was an exercise actually where I was comparing the Gillespie model to the ODE model. And so I loaded actually 2 copies of my original model. Notice I could load the same model string under 2 model names. So I have 2 copies of my model, which are independent of each other. One I solved with Gillespie and one I solved with the ODEs. And I can compare. I can ask the question, does the stochastic model make a significant difference or not? I think in the interest of time, I'm just going to leave that to you as a homework problem. Somebody got it to work. And so my suggestion was to do exactly this. And then you can change the initial value of A, 10, 100, 1,000. If you do that for the ODE, it doesn't really do anything. The scale of the y-axis changes. If you do it for Gillespie, it makes a big difference whether you have 10, 100, or 1,000. And when you overlay them the way I suggested, here's the Gillespie model, stochastic. Here's the deterministic model. And I'll just leave that for you for today. Exercise 5_1_4 does this. And I'd encourage you, sorry we don't have time to walk through it together here, but I'd encourage you to play with that. People are getting something here. If you run it for a long time, everything equilibrates. It's not so interesting. So the times between 0 and 300 are really the most interesting. These models run very fast. So, all right. And there are lots of homework extensions you can do exploring stochasticity. So this was different initial conditions, looking at the convergence. Here, if I had initially 10 molecules, they're very different. 100 molecules, they're a little bit different. 1,000 molecules, they're almost identical. And I appreciate the fact that we can't do everything in as much detail as people might want today. But I hope you get enough of a flavor today and in the afternoon, this morning and in the afternoon, that you have the material that you can then read Herbert's textbook or go back and look at some of the more detailed lectures from my courses or from previous years and fill things in. I'm seeing lots of very interesting stochastic results here. I tried to have every model written out for you so that you could download it in case you got stuck on some of these pieces. And there's some additional models in that folder that explore topics that we didn't have time to do today, like events. I guess the last thing I want to say is in the afternoon, instead of loading the antimony model into the global free-floating model, we're going to load it into individual cells. And then we're going to have each of the models inside each cell talk to the other cells. So we're actually going to have signaling between cells implemented this afternoon. That's a very powerful thing, which is not present in basic network models, the ability to have cells interact with each other. The other thing that'll happen is as cells come in contact or move around, the interactions will change. So you have dynamic coupling between networks.